Pollen pistil interaction. Pollen grains belongs to male reproductive organs of the flower. Pistil belongs to female reproductive organs of the flower. It is women of house. It is men of house. Pistil containing ovary, style, and stigma. The male reproductive organs like pollen grains deposited on the stigma. On this stigma, reactions between pollen grain and pistil called pollen pistil interactions. See pollen pistil interactions in angiosperms. See, at matured stage of the flower, carpel or pistil contains ovary, long style, and stigma. And at matured stage from anthers, pollen grains are released. The pollen grains are 3-celled stage, sometimes 2-celled stage. The 2-celled stage, 2 cells are generative cell and pollen tube cell. 3 cells are also present in the matured pollen grain sometimes. The 3 cells are male gametes, are also called sperm cells, and 1 tube cell is present with the pollen tube nucleus. See, at the matured stage, matured pollen grains deposited on the stigma is called pollination. Matured stage of flower, matured pollen grains deposited on the stigma is called pollination. In angiosperms, the pollination mechanism done by water, wind, insects, bats, snakes, like birds. These are the factors of the pollination mechanism. By these all factors, matured pollen grains are deposited on the stigma. See, during the pollination, different types of pollen grains, different species of pollen grains. Different species of pollen grains are deposited on the stigma. If the carpel or pistil is in matured condition, the stigmas are received pollen grains. See, different species of pollen grains deposited on the stigma. From this all types of pollen grains, only same species of pollen grains are germinated on the stigma. See, in angiosperms, mainly 2 types of stigmas are present. One is wet stigmas and dry stigmas. Whatever it may be, wet stigmas and dry stigmas are containing glands on the terminal part of stigma. By these glands, they released sucrose chemicals. The chemicals are spread on the over of the stigma. On this stigma, different species of pollen grains are already deposited by the pollination mechanism. From these all types of pollen grains, only same species of pollen grains are germinated, and remaining species of pollen grains disappeared or degenerated. This process is called pollen pistil interaction. This process is called pollen pistil interaction. See, pollen grains, same species of pollen grains absorbed sugar solutions on the stigma. They are developed into pollen tube. The pollen tube enter into style. It is one part of carpel or pistil. Long style is present in the carpel or pistil. Mainly in angiosperms, the styles are mainly 2 types. One is intercellular styles and halo-shaped styles. If intercellular spaces of styles are present in the style, the pollen tube reached or moves through the intercellular spaces only. through the intercellular spaces. Finally, the pollen tube reaches to the ovule. If hollow shaped styles are present in the angiosperms, the pollen tube reaches to the ovule through the hollow shaped structure. Finally, the pollen tube reaches to the ovule. It enters into ovule by three ways. It enters into ovule by three ways. See, here it is, the pollen tubes are reached to the ovule, enter into ovule by three ways. One is porogamy, second one is chalajogamy, third one is mesogamy. Porogamy means pollen tube enter into ovule through the micropylar region. Pollen tube enter into the ovule through the micropylar region is called porogamy. Example, paddy. In paddy, the pollen tube enter into the ovule through the micropylar region of ovule is called porogamy. The next type is chalajogamy. The pollen tube enter into the ovule. Pollen tube enter into the ovule through the chalaja is called chalajogamy. Pollen tube enter into the ovule is called chalajogamy. Pollen tube enter into the ovule through the chalaja called chalajogamy. And the casuarina is the best example for the chalajogamy. And mesogamy, pollen tube enter into the ovule through the integuments. See, these are the integuments. Pollen tube enter into the ovule through the integuments is called mesogamy. It is the best example, Cucurbitaceae family plants. Porogamy means pollen tube enter into the ovule through the micropylar region called porogamy. Chalajogamy means pollen tube enter into the ovule through the chalaja is called chalajogamy. Mesogamy means pollen tube enter into the ovule through the integuments called mesogamy. Porogamy examples are paddy. Chalajogamy examples are casuarina. Mesogamy examples are Cucurbitaceae family plants. Finally, by three ways, pollen tube enter into the ovule. It reaches to the micropylar region. Finally, the pollen tube reaches to the micropylar region. Means, if the pollen tube enter into the ovule through the micropylar region, if the pollen tube enter into the ovule through the chalajal region, if the pollen tube enter into the integuments through the chalaja, through the integuments called mesogamy. If chalajogamy, mesogamy, pollen tubes are finally reached to the micropylar region of the ovule. See, at the micropylar region of ovule, pollen tube finally reached to the ovule, enter into ovule by three ways: porogamy, chalajogamy, mesogamy. If the three ways of pollen tubes finally reach to the micropylar region, at end of these micropylar region, pollen tube is received by the synergids. The pollen tubes are received by the synergids because the synergids are containing filiform operators. The filiform operators are present on the synergids. They are finger-like structures. They are present on the synergids. That synergids are mainly helps in receiving of pollen tube during the fertilization. The synergids of filiform operators received pollen tube and it entered into the synergid, and the pollen tube terminal part is ruptured. It released two male gametes. From these two male gametes, one male gamete fused with egg cell to form diploid zygote. This fertilization is the first fertilization. It is also called true fertilization, and it is also called syngamy. The true fertilization or first fertilization or syngamy first identified by Strasburger scientist. And the second male gamete or second sperm cell released into the embryo sac. It reached to the center region of embryo sac. It fused with the secondary nucleus. It fused with secondary nucleus to form triploid cell. The triploid cell is also called PEN, primary endosperm nucleus. It is also called double fertilization. Double fertilization identified by Novachin scientist. See, the first fertilization identified by the Strasburger, and the double fertilization or triple fusion is identified by the Novachin scientist. See, it is the pollen pistil interaction with fertilization. See, pollen grains are deposited on the stigma. Only same species of pollen grains are germinated by the absorbing of sugar solutions, and they develop into long pollen tube. The pollen tube contains two male gametes called as two sperm cells, and end of the pollen tube nucleus is present. Finally, the pollen tube enter into the ovule by three ways. One is porogamy, chalazogamy, mesogamy. Whatever it may be, the pollen tube finally reached to the embryo sac, and it means the pollen tube received by the synergids. The synergids received pollen tubes, and pollen tube terminal part is ruptured, and the pollen tube released two male gametes, two equal male gametes. From the two equal male gametes, one male gamete fused with egg cell to form diploid zygote is also called true fertilization. It is also called syngamy. It is first identified by Strasburger scientist. See, another male gamete reached to the center region of embryo sac. It fused with central cell or secondary nucleus to form diploid zygote. To form PEN, primary endosperm nucleus. This process is also called, this fertilization is also called double fertilization. First fertilization with egg cell to form zygote. Second fertilization with secondary nucleus or central cell called double fertilization. True fertilization or syngamy identified by Strasburger scientist, and double fertilization or triple fusion. Triple fusion means in secondary or central cell, two nuclei are present. The two nuclei, including male nucleus, the three nuclei are fused to form PEN, primary endosperm nucleus. So this process is also called triple fusion or double fertilization. It is identified by the Novachin scientist. Finally, in egg cell, the egg cell convert into zygotes. See, here it is discussed post-fertilization changes. Post-fertilization changes or events. See, children, here it is post-fertilization. See, parts before the fertilization in flower, transformation after fertilization in flower. See, we observed the sepals, petals, stamens, style, and stigma. These are usually fall off from the flower. And ovary, after fertilization, convert into fruit. Ovule, after fertilization, it convert into seed. Egg cell, after fertilization, it convert into zygote. Funicle, after fertilization, it convert into stalk of the seed. Micropyle of the ovule, after fertilization, micropyle of ovule of the seed, micropyle of the seed. It is mainly helps in receiving of oxygen and water. And nucellus after fertilization, it convert into perisperm. It is the nutrient tissues. Outer integument of ovule convert into testa, outer coat, seed coat. And inner integument, tegmen, it is inner seed coat. Synergids degenerated. Secondary nucleus convert into endosperm. Antipodals degenerated. After fertilization in ovule or in pistil, we identify zygote. It convert into embryo, and PEN, primary endosperm nucleus convert into endosperm. These are the post-fertilization events in the angiosperms.